The title of the topic that I'm presenting today is in vitro characterization of 5 potential antigenic proteins with specific immune reaction to tuberculous mycobacterium species. Let me first start with a brief historical overview about tuberculosis. Tuberculosis is actually one of the very old diseases that have been known on Earth. Like some literature say it's more than 10,000 years old. Some interesting literatures characterize the DNA of Mycobacterium tuberculosis from Egyptian mummies belonging to very long ancient time ago. Actually, it is believed that tuberculosis is originally a cattle disease, a disease of cattle, from wild cattle. And it transferred from cattle to human being at the time when the human being first time domesticated cattle, like 10,000 years before. And it was the time when the disease started to infect the human being and become a human disease. So tuberculosis is believed to be a cattle disease, and at the time of domestication of the cattle by human being transferred to human being, causing tuberculosis in human being. And some evidence that supports this hypothesis is that the microorganism responsible for tuberculosis in animals is called Mycobacterium bovis, can infect both animal and the human being, while the microorganism which is underwent evolution over the time and infect only human being cannot infect cattle. So that's why they believe that Mycobacterium bovis is the origin of tuberculosis, and then it moved to human being and evoluted into the Mycobacterium tuberculosis. So in WHO report, one of the oldest reports, they speculated that during the first 20 years of the third millennium, we will have a large number of people who are infected with tuberculosis, and they estimated this number close to 1 billion new cases of patients or people will be infected with, newly infected with tuberculosis, from which they speculated 200 million will show symptoms, active disease, and from which 35 million will die, mostly in the developing countries. Of course, Mycobacterium tuberculosis will be responsible or is responsible for a large number of this speculated number, but unknown and the potentially important proportion will be caused by Mycobacterium bovis. So controlling the disease in human being cannot go successfully without controlling the disease in animals. That's why control the bovine tuberculosis is very important and very crucial for treatment and controlling the tuberculosis overall among human beings and animals. The general strategy for controlling tuberculosis in animals depend on what's called the test and slaughter strategy, using the skin test based on a reagent called the tuberculin that provide protein derivative of tuberculin, which is depend on delay type hypersensitivity reaction shown on the skin. And based on this test, all the suspected animals will be tested by the skin test, and the positive reactors will be slaughtered. So that's the only way to destroy the disease and eliminate the disease among animals and get rid of a source of infection to human beings. So tuberculin have been used for decades as an asset for the control of tuberculosis among animals. And the problem that we facing with tuberculin is that tuberculin, when used as a skin test, 72 hours after injection intradermally, we get the readings or results, and the result is a positive result or a negative result. Negative result meaning that we don't have a disease, it's okay, but if it is positive result, in this case the animal is slaughtered, as I said earlier, and after slaughter we're looking for lesions. If there's lesions, the animal will be destroyed, either partial or complete destruction. But in some cases, after slaughtering the positive reactors, we didn't find any lesions. And this case were assumed to be a false positive cases of the tuberculin. And this represents roughly from 10 to 15% of the positive reactor cases have been confirmed to be false positive case. This have a very bad impact, both economically and epidemiologically, and can represent a risk for the control program of TB, because in many countries, like in my country, I've seen this by myself, in many farmers, especially the owners of big farms, try to escape the testing, because they know that some of the positive reactors will be false positives, and you'll discover this after destroying the animals. So they lose a large part of their population of their farms, and so they try to escape the testing, either by doing it in the wrong way, or getting the animals that have to be proved positive and have to be destroyed, and they sell it in the markets to just get some benefits, not to destroying them. So having this problem with tuberculin can affect the control program of tuberculosis. This false positive results is actually, or can say it is a problem in the specificity of the test, is attributed to presence of sharing antigens component in the tuberculin test. The reagent we are using, the tuberculin, having some sharing antigens that shared between the tuberculous mycobacterium, which is the main cause of tuberculosis, and other non-tuberculous mycobacterium, or what we call it environmental mycobacterium, which didn't cause the disease, but can sensitize the animals and make the animal positive reactors for tuberculin. And this is the main cause of false positive results. So the aim of our work was to implement the biotechnology to improve the skin test assay, to keep the skin test assay as a simple and a cheap field test, and to be more suitable and more visible for the control of the bovine tuberculosis. Our hypothesis beyond this is that the currently used tuberculin complex, although it's not highly specific, it does contain protein unit fractions only specific to Mycobacterium bovis, among other cross-reactive proteins. So the goal of our project was to identify those specific proteins to be used for development of a more reliable skin test to improve the control of the bovine tuberculosis. The overall protocol of our work started from the production of tuberculin from standard strains of Mycobacterium bovis, and subject the tuberculin for fractionation using the HPLC. Then preparing guinea pig models for both tuberculous and non-tuberculous Mycobacterium. And then the obtained fractions out of fractionation will be tested by in vitro evaluation using both the lymphocytic proliferation assay and the gamma interferon assay, by testing the sensitized peripheral blood monocytic cells from the guinea pig models of both tuberculous and the non-tuberculous animals, and compare this with the result of the conventional PBD or the conventional tuberculin, in addition to the negative control groups. At this level, after in vitro evaluation, the selected fractions or selected proteins that prove to be specific and antigenic, or antigenic and specific, will be subjected to a second level of evaluation, which is the in vivo evaluation by skin testing in the guinea pig models. And the selected proteins out of this phase will be subjected for field evaluation using cattle models. Okay, so we start first by sensitization of the laboratory animals, the guinea pig models, and for this purpose we used 5 different reference mycobacterial species, in addition to the Mycobacterium bovis, the 5 strains, the Mycobacterium tuberculosis and Mycobacterium bovis. Both of them are tuberculous Mycobacterium, in addition to the non-tuberculous Mycobacterium represented by Mycobacterium intercellulare, Mycobacterium avium, kansasi, and fortuito. We sensitized the animals, or we prepared our guinea pig models by intramuscular injection with a half ml of 2 milligram per ml of heat-killed strains. And as I said, we prepared 3 different groups. First group, model is the tuberculous infection model, and composed of 2 groups, one for Mycobacterium tuberculosis and one for Mycobacterium bovis. And then 4 different groups representing the non-tuberculous infection models, with using the 4 different non-tuberculous mycobacterial strains, and the negative control group. Then the second step was to prepare the culture filtrate and the tuberculin purified protein derivative from the reference Mycobacterium bovis strain. And we briefly did this by cultivating the Mycobacterium bovis on enriched liquid media for 4 weeks, and then put it in a synthetic certain media for additional 7 days with shaking. Then we collected the supernatant from this culture and divided into 2 parts. One part were inactivated by autoclaving and used for production of what is called the heat-killed purified protein derivative, which is the conventional protein, the conventional mean tuberculin. And the other part of supernatant were subjected not to heat inactivation, but subjected to sterile filtration. So we produced what's called the sterile filtrate purified protein derivatives tuberculin. And this one, which is the one which we used for the fractionation, to be used for the evolution study later on, because using the conventional one, which is the heat killed, cannot be fractionated because of the heat inactivation of the tuberculin affects the protein content, and so it is very hard actually to make fractions out of this and to separate it in purified form. Then both the obtained fractions, the obtained fractions out of this step will be evaluated both in vitro and in vivo and compared with the result of the conventional tuberculin. So we start by first subjecting the sterile filtrate tuberculin into SDS page analysis to see its protein, to get sure that we're having different protein contents. Then we subjected to HPLC, reverse phase HPLC analysis. And initially, we were able to, using different run conditions by changing the pH and changing the concentration of the reagents, we were able to develop or to generate 16 different peaks of or fractions of protein. So at this stage, after getting this 16 different fractions, to get sure that this fractions is a pure protein, we subjected for SDS page analysis, and we found that all of them were purified protein except 3 fractions were still multiple proteins. So we resubject them for refractionation using shallower gradient during the run. So we were able to fractionate them and to purify or to separate them in pure fractions with pure proteins, which was confirmed later on by the SDS page analysis. So now we end up with having 21 different protein fractions out of the fractionation step. Then we start the in vitro evaluation stage or study of these obtained fractions. And for this purpose, we collected whole blood sample from the guinea pig models, and we, using the lymphocytic separation media, we equal volume from both blood and lymphocytic separation media. We added the blood over the lymphocytic separation media, making the centrifugation, and we were able to separate the buffy coat layer, which including the the blood cell or the lymphocytes, which were collected and used for the conduction of the in vitro evaluation study, which was, as I said earlier, both the lymphocytic proliferation assay and the gamma interferon assay. So we used cells, incubate the cells with the protein fractions collected after fractionation, and also as a control, we compared this result with the result of incubating this sensitized cells and unsensitized cells from different groups of guinea pig models with the conventional tuberculin. So for evaluating the antigenicity of the selected or the obtained fractions, the 21 fractions, we used peripheral blood monocytic cells or lymphocytes from sensitized guinea pig models with tuberculous Mycobacterium species to see which fraction can induce a positive result in terms of lymphocytic proliferation or gamma interferon production. And to evaluate the specificity of this antigenic fractions, we used the same step, but in this time we used the lymphocytes from sensitized guinea pigs with both tuberculous and non-tuberculous Mycobacterium. So to test the specificity, the good fractions should give only fraction or give only reaction with the sensitized lymphocytes from tuberculous mycobacterium and not from the non-tuberculous mycobacterium guinea pig models. The results that we obtained from the in vitro evaluation study showed that 12 different fractions can or showed comparable result to the conventional purified protein derivative of the tubercline. And in terms of specificity of these antigenic fractions, when we used the guinea pig or the lymphocytes from guinea pig models of both tuberculous and non-tuberculous mycobacterium, we were able to define only 5 fractions that were showing reaction only with tuberculous mycobacterium and not with the non-tuberculous mycobacterium. So these 5 fractions here, fraction 1, 2, 3, 4, and 5, showing only comparable reaction to BBD only with the Mycobacterium tuberculosis, Mycobacterium bovis models, and not with the other non-tuberculous mycobacterium, while the BBD showing reaction with all the tuberculous and non-tuberculous mycobacterium, and that's why it's having the specificity problem. So we end up with 5 fractions that show in vitro, showing a very promising results. When we try to repeat this result in vivo using by skin testing on animal models, actually we fail to get comparable results or comparable reaction to that of the purified protein derivative, meaning that purified protein derivative, and right here, maybe it's and right here, this is the reaction from skin testing with purified protein derivative, but here this is representative samples from the 5 fractions that we obtained. It didn't show comparable results as we obtained by the BBD. So we failed at the level of the in vivo reaction. Although at the vitro, in vitro reaction, we can specify these 5 different fractions having antigenic and specific reaction comparable to purified protein derivative of tuberculin, but in vivo, when we use it as a skin test reagent, it didn't show the same results. Okay. Then we go one step further. We make molecular characterization of this selected proteins. First, to get sure from the purity of these proteins, we run it on STS-page analysis, and to know what is exact molecular weight the range of molecular weight we have, and we end up with a molecular range from molecular weight range from 5 to 25 kilodalton. Then these selected 5 fractions were subjected to N-terminus sequencing of this protein by Edman degradation, and we use this sequence to make a cluster analysis for protein using the database of Mycobacterium tuberculosis proteins, and we were able to identify the sequence of the 5 proteins and with this excision numbers from the gene bank. Then we identified the open reading frames of these genes were characterized, and currently we are working on the cloning of these genes for mass production of the corresponding recombinant protein. Okay, so the conclusion of the results that we obtained from our project so far, that we were successful to identify 5 different antigenic proteins that showed specific in vitro delay type hypersensitivity reaction comparable to the tuberculin only in Mycobacterium tuberculosis model and not in non-tuberculous Mycobacterium model. However, as I said, none of these proteins succeeded to produce any delay type hypersensitivity reaction comparable to that of the conventional tuberculin when used as a reagent of skin testing or in vivo evaluation. The fact that tuberculin, the conventional tuberculin, is a protein complex with many pro-inflammatory stimulus can explain this issue of sensitivity. It turned out in our suggestion to this results that the failure of these reagents to produce in vivo reaction is a matter of sensitivity. The sensitivity of purified or conventional tuberculin, which is composed of multiple protein complex having pro-inflammatory agents, having a stronger sensitivity than single protein fractions, each one injected by itself. So the complex nature of tuberculin could have support the notion that these proteins could have represented potential candidates for skin testing, not individually, but in a cocktail. So in addition, inclusion of some synthetic bacterial lipoproteins as an adjuvant in the proposed specific antigenic protein cocktails could provide the assumed required pro-inflammatory stimulus and enhance the delay type hypersensitivity reaction which is required to be shown by these proteins. Therefore, currently we are working on the cloning and the production of this antigen as a recombinant protein to be used in different combinations with or without adjuvants to be tried and see if we can improve its sensitivity after we proved its specificity. We now working on the sensitivity. Hopefully we can get a good result at this stage. By this, I conclude my presentation. And finally, I would like to acknowledge that this work was a part of a project funded by the National Science, Technology, and Innovation Plan by King Abdulaziz City for Science and Technology, Kingdom of Saudi Arabia. Thank you.